just give it another minute, just in case.
Great lecture today.
It's quiet than in class, right?
Or I guess why would that person address it?
Jim. How did you fumble today? Huh? What happened? Sorry, scared with that board.
Oh,
No, we didn't look at that yet.
No, we haven't. It's nothing related to my
But also,
I just,
through my.
That's right, everyone. So let's get started. Thank you, everyone, for coming. It's really nice to see you. Today, we're very excited about having our very own Dr. Andrew Conith give our annual Darwin Day seminar. So this is something now that's been going on for a few years. If Dr. LaMontagne were here, she'd tell me exactly how many years it was, because she co-started it. But we're very excited. So Darwin Day is an event that's celebrated at institutions really throughout the world, throughout the United States and other places. And it's a general celebration of science, and it's more specifically a celebration of really one of the most important scientific ideas that we've had. The notion that all species are connected, that humans are part of that tree of life, that species can change over time, and that there's a mechanism that's pretty straightforward, natural selection, that can explain the process of adaptation, that fit between organisms and their environment. So evolution is fundamental for understanding the natural world, but it also matters for many other perspectives, including for human disease. Where do new pandemics come from? They evolve usually from viruses that occur in other animal species. Why does your back hurt when you get older? Right, our knees, we have problems with our knees. Autoimmune diseases are on the rise throughout richer economies. Problems with mental health. So there's a lot of things that are evolutionary history, what humans actually evolved to do, and the mismatch between how we live our lives today can account for many of the health problems we're facing as a society. Increase in type 2 diabetes. I'm a victim on that front. I'm a terrible victim. But anyway, so we've been doing this for at least, it's been over 10 years now. And we're very excited to have our professor, Andrew Conith, to give our seminar today. Dr. Conith is originally from Derby in England. He got his bachelor's in science in zoology from the University of Leeds, also in England, where he worked on a thesis project on recombinating the sine oculus homeobox gene, CHEH34. So he's done some work on homeobox genes with his advisor then, Dr. Ian Hope. Then he went on, and someone that's very intellectually curious, obviously, he went on to do a Master's of Science in Paleobiology, and at the University of Bristol in England. And his thesis project was on the taxonomic diversity of the stem mammal. Exactly. Exactly. He was co-advised by Dr. Pam Gill and Dr. Emily Rayfield. Then he came to the US and he got his PhD in organismic and evolutionary biology in 2017, where he worked on a project looking at the role of integration in shaping the diversity of mammalian jaws. And he worked on that project with Dr. Elizabeth Dumont, and then finally he went on to get a postdoc, also at the University of Massachusetts at Amherst. And that project was looking at the genetic and developmental mechanisms that underlie craniofacial variation, where he had his most brilliant advance. He moved on to the world of fish. Yeah, and worked with Dr. Craig Albertson. So we're very happy to have you here, and I will let Dr. Conith take it.
Thank you everybody for coming. It's kind of interesting, so February 16th. So I was in this exact position on February 8th of last year, doing my job talk. So it's like I'm getting like a lot of deja vu right now. Not as stressed as I was that day, but still, we'll go through some good stuff. Okay, well, I'm super excited to kind of talk about Darwin, kind of get a sense about what could Darwin contribute today? If he could rewrite the origin of species, what might he put in? What examples would he want to use? Right? And so, if we think about where he was, you just heard that I was from Derby, so I was stuck up in kind of the Midlands of England right there. Darwin was also from England, and he got the opportunity to serve as kind of the resident geologist on this trip, on the HMS Beagle. And so the main goal of this was just to survey South America. And at the same time, he got a lot of money to look at some of the local flora, the local fauna, and then send them back to collectors who wanted them. And so following that trip and being on the Beagle, he wrote this book. So The Voyage of the Beagle. So he talks about finches in this. He talks about kind of his adventures across the world, but doesn't really start to get in any depth about evolution or anything like that. And so his time on the Galapagos, I think, really shaped some of his initial theories, but he kind of kept them quiet. Right? So we saw variation in beak shapes. How did that variation come about? Why are they so different? Right? And so we started to get a sense about, did they all come from the same common ancestor? Okay. And then in 1859, he published this. So he'd had a handful of conversations with Wallace, and this came about. It's 13 chapters, a huge synthesis of all of his ideas surrounding evolution by natural selection. And so he has all of these kind of notes, and the only figure that is in that book is this. And so this kind of gives you a sense about this branching pattern, evolution as a branching pattern. And so he went forward. There was a handful of other editions, but the 6th edition, this one here, this was the last one that he did. It was a little bit smaller, and the idea was that he wanted more people to read it. He wanted as many people to read it as possible. There's this, at the time there was this intense class system in England, and he wanted to get as many people to read it as humanly possible. And so this really brought the cost down. If you want this book today, though, it's like $1,800. So it's kind of pricey. I don't think many people could just afford that anymore. But it got me thinking about what did Darwin contribute and what have we contributed since? Right? So here's Darwin's contributions in yellow here. And then there was kind of 2 major waves following that, where huge additions to the theory of evolution by natural selection came about. So the first one kind of came about in the early 1900s, and it was this idea called the modern synthesis. So it merged the ideas of Mendel with the ideas of Darwin. And there was a handful of people that were involved. These two, Meyer and Dobzhansky, were probably the 2 big ones. And I think one of the interesting things about this period in time, so this was during World War I and World War II. So you had a large number of people who were engineers, like really brilliant mathematicians who were working on figuring out how to build planes, build new machines. And then following the war, you had all of these unemployed engineers that were like, well, what are we going to do now? And so a lot of them put their minds together to think about population genetics and start to think about some of these rules of evolution in mathematical terms. And then we had what's kind of been termed as the extended evolutionary synthesis. So this is a little bit more recent. And so this is in blue here: genomics, evolvability, modularity, plasticity. All of these new kind of fields that are coming about over the last 30 to 40 years or so, that are building on these original ideas put forward by Darwin. And so these are probably what I consider maybe like the three major players. So Pigalucci, Hoekstra, and Müller. And so they work in kind of genomics, genetics, trying to understand the genetic basis of a lot of these different traits. Let me see. Okay. And so it got me thinking about, if Darwin came back, what would he do? How would he write the 7th edition? What would he want to add to the origin? Right? And it kind of got me thinking, because I saw this episode recently of Doctor Who. And so in Doctor Who, the Doctor goes back in time to meet Van Gogh. And at the very end of the show, what he wants to do is he wants to bring Van Gogh to the modern day. So Van Gogh was obviously super depressed in this time. He basically made no monies from his paintings, and he essentially thought, everybody hates my work. And so what the Doctor wanted to do was bring him to the present day. And he brings him to Paris, and he shows him one of his galleries where all of these different paintings are. And Van Gogh sort of hides in the shadows and just like listens to people talking about his work. And it's this kind of really poignant moment where he kind of gets the realization that his art was amazing and was incredible. And so I kind of thought about it in the same sort of sense. If Darwin was able to travel in the TARDIS to the modern day, what would he want to do? What would he think about how people perceived his work now? And so with that, I want to go through almost like a series of chapters from the origin and put them in today's context. So the first chapter, variation under domestication. So this isn't my work. I'm going to be kind of pulling from a lab from Utah, the Shapiro lab. But Darwin loved pigeons. And pigeons were this great example of artificial selection, right? He was able to prime the readers of the origin to why variation in traits is so important using selective breeding. I then want to get into chapter 4. So this is his massive chapter on natural selection. And in that, I want to talk about evolvability. So the ability of organisms to evolve. And so I'm going to talk about my favorite groups there, so cichlids. Can't avoid a good cichlid. And then the final one is chapter 5, so law of variation. And so this is another thing that I think Darwin would be really interested in looking at, so developmental modularity. And I'm going to be talking about both cichlids in that chapter, and then also a little bit about mammals. Okay. Right. So pigeons. So I knew that pigeons were like pretty wild, but some of the ways that we've been able to tweak aspects of their phenotype is pretty nuts. They exhibit a whole load of different, obviously, feather phenotypes. They exhibit a whole load of different behavioral phenotypes. And many of them have really simple genetic bases. Okay? And so we've been able to figure this out from the worlds of genomics and then just straight up Mendelian genetics. Okay? So this is what pigeons look like, or domesticated pigeons looked like in Darwin's time. And so he knew that it was going to be a really powerful way to talk about some of his ideas, prime the reader for natural selection. People were exposed to artificial selection all over the place, in livestock, in cats, in dogs, in birds. So it was pretty commonplace. It was easy to use it as an analogy for natural selection. Right? So he wanted to basically show that all of this variation in pigeons had just built up over a few hundred generations. And so what happens if you increase that time scale? Right? Is that how the diversity of life has arisen? Right? So one of the first things he did, and I think this was pretty commonplace at the time, was that he had a pretty good sense that there was a single common ancestor for all of these different pigeon breeds. Okay. great as are the differences between the breeds of pigeon, I am fully convinced that the common opinion of naturalists is correct, namely that all are descended from the rock pigeon. And so I think one thing that Darwin would be interested in is starting to play with some of the genetics, get a good sense about how do we gain all of this variation in these pigeons. And so I'll go through a couple of different examples, pigment and then the crests. So here's your wild type, like rock dove, rock pigeon. And they have these different pigment types. So you have the wild type one here, and then what they've named spread. Right? So this is where the pigment is evenly distributed over the entirety of the feathers. And it has a really super simple genetic basis. It's just a single gene with two different alleles. And so if you are homozygous or heterozygous for the spread allele, then you're going to have that kind of bluish coloration. If you're recessive, you're not. You're going to kind of be a little bit grayer. Okay? And so you have this dominance idea. This was something that kind of Mendel was talking about. Okay? But it's a little bit more complicated than that. There's another gene. So here's spread, and then linked to it fairly weakly, but still linked, is something called pattern. And so there's this epistatic interaction between these two different genes. And so in order to perhaps get a sense about what these different birds can look like, you can cross them. Right? So what happens if we cross this pigeon here with the recessive spread genotype with a couple of these different pattern genes, and then the homozygous for the spread genotype with these other different pattern genes. And so the parents can exhibit these three different alleles. They can be this T-check, where it's kind of dark, check, where it's kind of, it actually looks like little triangles, and then bar, where it's all striped. Okay. So when you breed them together, you would get something like this. So this is your T-check, this is your check, and then these are just your, the spread allele there is masking everything else. So we'd be able to use Mendelian genetics to get a sense about how some of these traits are arising in pigeons. And so this is what they look like. Okay. So this is that T-check. So basically the entirety of the wings are all kind of blackish, and they have these patterned in the checked, and then this is where the spread just totally masks everything. And then the crest. So the crest is really wild. So the crest is essentially the presence of this little tuft on the back of their heads. So they stand up on the back, and a single gene, again, controls the presence or absence of a crest. So if you are recessive for this, then you'll exhibit the trait. If you are homozygous or heterozygous for the no crest, then you will not have it. Really simple genetic basis again. And so what they're able to do here is actually get a sense about what gene is controlling this and how has it changed. And so the specific gene is this FM receptor. And it's all just due to a change in a single nucleotide. And so by just tweaking that single nucleotide, they tweak the amino acid sequence, and it essentially controls polarity of the feather development. Okay. Oh yeah, it's just that. Okay. So this is, if you look at the back of the heads of these pigeons, a crested version versus an uncrested, this is an in situ hybridization, basically, where you're looking at where the genes are expressed, so where this gene is expressed. Those little dots there, that's called a feather placode. So that's where feathers are going to grow from. You'll notice that the direction of the most expression of those genes is all facing down. Whereas in the crested ones, it's kind of a bit mixed up around here, but then it's all facing up. And so this gene essentially impacts the polarity of how these different feathers are going to develop. Are they going to develop down, or are they going to develop up? But obviously, it can get even more complicated. Right? So the presence or absence of a crest is just controlled by this single gene, but then you can modify it massively. So how much crest are you going to have? Often what they've done here is that the neck essentially extends, and so it kind of accentuates the whole trait. But what I hope Darwin could take away from some of this is that the combination of both these kind of simple and complex genetic traits can lead to pretty wild differences in phenotypes. Okay, so let's tackle natural selection. So I want to look at evolvability in this case. So the ability to produce adaptive phenotypic variation. Okay. So obviously when Darwin went to the Galapagos, he saw all these different finches. These finches differ massively in their beak shapes. And it's likely driven by diet, right? You have these ones for seeds, you have these ones for insects, these ones for cacti. And so form is following function in this case. Right? The form of the beaks is following the function of what they're eating. Okay? And it's just classic Bio 192. There's variation in the trait, there's a selective force on that trait. Some survive, some don't. And those that survive are able to pass that trait on to the next generation if it's heritable. So that brings me to some of my stuff. So I'm mostly interested in the vertebrate head. It's this really interesting, shape. Right? It has to do a whole host of different things. It has to be able to communicate. Often there's going to be sexually selected ornaments that are on the top of the head. It has to be able to eat. It potentially has to be able to breathe and respire. So all of these different trade-offs are all going to impact the final shape of it. Okay? So how does it work? How does it develop, yet still being able to maintain all of this functionality? Okay. And so a lot of the ways I go to deal with this question is to look at cichlids. So cichlids are this wildly morphologically diverse group of vertebrates. Much of my work focuses on the African radiation, or multiple radiations in Africa. And I really hope that Darwin would love these kinds of fish. So this is something he said. So in the general economy of any land, the more widely and perfectly the animals and plants are diversified for different habitats of life, so will a greater number of individuals be capable of supporting themselves. So that just gives you, so he's kind of thinking about form function, how you can get different organisms moving into different niches. Right? And so I think that cichlids really kind of tick that box. They've been able to invade a whole host of different aquatic niches. And so to bring those two things together, so I'm interested in the shapes of the heads in cichlids. So how did that diversity arise through natural and sexual means? And then I try to take this sort of multi-hierarchical approach. And so today I'm mostly going to be looking at this transition. So the morphology to performance. So how does form relate to function? How do those functions operate in the environment? So these are two different fish that I've done the majority of my work on. So this is a more sort of specialist feeder here. So this is called Labiotropheus. So it sits close to the rocks. It has a really robust jaw, a really robust head, and it is a specialist algal scraper. It essentially just kind of sits and grazes along the rocks, kind of slowly plucking at the filamentous algae that reside on those rocks. And it's really, really efficient. The lake it lives in, Lake Malawi, it has this ubiquitous distribution. It's everywhere in the lake. And there's maybe about 3 to 5 species or so. And so this is one group that we're really, really interested in. How is it so successful? What makes it a successful cichlid? And then this is the other genus that I'm typically interested in. So this is Tropheops. This is more of a generalist. it feeds off the rocks, it'll feed from algae, it'll also feed on benthic invertebrates, it'll feed on pelagic invertebrates, just does a little bit of everything. It does a bit of everything, but it's not great at everything. A lot of the behaviors you'll see here where it kind of does this like kind of nipping and twisting are really energetically expensive. And it doesn't have a ubiquitous distribution like maybe Atrophius does. So why? Like, why are they so different? And so I've been working with Sarah, one of the master students here, and she's been trying to take a look at Trophiops, trying to look and characterize variation in Trophiops. So Trophiops are super cool. We were able to scan some of them at University of Chicago. So here's their CT scanner. You plop a little set of samples in there, and as they come out, they look a bit like this. So you can kind of see it's just X-rays. I've got them arrayed in like 3 different rows here. It's probably about 3 to 4 specimens in each of those rows. And then that's what it looks like on the inside. So you have these 2 different beam lines. This is the detector. So basically, X-rays are being blasted up against your specimen. Some get absorbed, some get reflected. The detector picks them up and can build a 3D picture of your specimen. So this is a cichlid skull, and so its eyes would be here. This is the front here. So the rest of its body would be around here. And so what Sarah's been doing is trying to characterize the shape of their heads, trying to understand these form-function relationships. You first got to be able to get form. And so you characterize the shape of them, and one of the ways that you often analyze them is with something called principal component analysis. So this basically distills all of your different data down to a series of axes that best explain shape variation. So what we're going to see on this plot is a series of points. The closer those points are together mean that those 2 individuals are more similar in shape. The further away they are, the more different they are in shape. And there's 3 different groups, 4 different groups actually. Okay, so those axes here represent some kind of change in shape. So at this end, across this and across this, I'll show you what their shapes look like. So this first axis basically shows you change in depth, width, length. And then across this axis, it basically shows you change in a bone called the vomer. So this is a bone that sits right at the front, right at the tip of the snout. So this is the major change along this axis, PC1. And then this is the major change along PC2. Okay. So what we want to do now is try to get a sense about why are they different? Are there any other traits that we can kind of pull out that give us a little bit more information about how that form relates to that function and that performance? Okay. So one of them is this angle here. So this is the vomer, this little bone here. In some fish, it's really essentially 90 degrees. And then in some fish, it's a lot more obtuse. And what we're thinking is that this is associated with how they feed. This here, this is Labiotrophius. This is Trophiops. Trophiops can live in kind of deep environments, or it can live in shallow environments. If it lives in shallow environments, it typically exhibits shapes more similar to Labiotrophius. But it will vary in that, it can vary in that vomer angle, it can vary in width, length, and depth. So we want to get a sense for this whole spectrum of changes. How do you reach this phenotype, right? The phenotype being this vomer angle, differences here. Okay? So you have a suite of genes, they're modified by development, but they're also modified, or they're also impacted by performance. So how are they actually using these traits? Right? And this is one of the things that Sarah's going to be checking out soon. It brings on the most severe struggle between the individuals, whether of the same or of different species which subsist on the same kind of food. Right? Labiotrophius and Trophius often eat very similar things. One is an obligate algal scraper, and one will kind of just pick at algae when it needs to. So we'll be seeing some ways that they're able to be, one group is able to be much better at doing it than the other. And so there's clearly differences in morphology, right? So these ones eat a huge amount of filamentous algae. This is a species called Melandia. This will eat a bit of algae, but it'll also mostly eat invertebrates. Okay, so one of the things that Sarah's going to be doing is basically taking a look at these VOBA angles, and then she's going to be seeing and recording their performance, trying to get a sense for, is there any association between the performance and the morphology? What angles are they feeding at? How efficiently are they feeding? This is a like an algal map where you can see how the fish are actually biting onto it. So these are little tooth marks. So does form inform function and performance? Okay. So for the final chapter then, I wanted to go through these laws of variation. Right? So hopefully Darwin is interested in evolvability, and hopefully he's also interested in modularity. Okay? So modularity is the strength of association among different suites of traits. So how correlated are these different traits? Okay? And so for this, I've kind of used both lobed-finned, like us, so we're Sarcopterygians, and ray-finned fish. Right? So cichlids, and then I'm going to use some mammals to do this as well. But still, they both have heads, and so I like them. Okay. And so you have this effect, right, where you have some kind of suite of genes that will go in to pattern some kind of phenotype. But the problem is that you don't get this one-to-one interaction. It's got to go through development. So development is going to kind of tweak that to some degree. And so when it starts to tweak it, you're going to start getting suites of traits that are going to be more associated and interacting more often than other suites of traits. So how might that come about? Okay. So let's imagine this scenario where you have a muscle and a bone, they're developing at this interface. What's happening? Right? So let's say that you have these progenitor bone cells, and then a muscle starts to grow. And so this muscle is going to be exerting some kind of force on that suite of bone cells. And so as that force increases, there's going to be a bit of give and take. So the muscle is going to get larger. It's also going to have impact on the underlying bone. And so a suite of different genes are going to be expressed, a suite of different morphogens are going to be expressed, and you're going to start to see this association between those two traits, the muscle trait and the bone trait. Okay. And so what this means is that you can have changes to the muscle. So some change to a muscle gene, maybe it increases the volume or decreases the volume, that can have an impact on how the bone looks, even though you've got no similar changes occurring to the bone. Okay. And so we were able to demonstrate this in a group of zebrafish. And so we played around with this gene, this ADAM12. So this is a gene that's involved in ligament formation. And so this is the wild type here. So this is the ligament making a connection to the coronoid process. So this is a process on the jaw. And so what ADAM12 does is if you don't have a decent copy of that gene, the ligament is really, really reduced. And because there's interaction between the ligament and the bone, there's way less bone deposited in the coronoid process of those fish that don't have a decent copy of this gene. Okay. So this is an example about how you can tweak something to like a soft tissue gene and impact a hard tissue gene, impact a hard tissue structure. Okay. So what if you started to impact something at the interface? So that has an effect on both bones and muscles. Well, it's going to impact them at the same time. And so you can get these really quite distinct effects. So this is a mouse that doesn't have a working copy of PAX9. Mice normally have these two processes. coronoid process, the angular process. This is where the jaw joint is, just here. If it doesn't have a working version of that gene, it just totally loses those processes. So PAX9 is being secreted at the interface. It's regulating that coordination between the muscle and the bone. So how might we measure it? Well, we could start to measure these different traits and get a sense for how associated they are. And it's just a simple correlation matrix, essentially. So as you start to get a sense for how associated each of these different traits are, you'll start to get a sense for which ones are more associated. And you'll start to build this pattern up of suites of modules. So suites of traits that are more associated with each other than they are to the greater suite of global associations. Okay. So to kind of put it into the same framework, these structures can have this this scenario basically where you'll start to get a group of traits that all start to perhaps evolve together, and another group of traits that can evolve independently. And so we term this developmental modularity. And so I want to look at developmental modularity in mammals, and then I'll look at it kind of briefly in cichlids. So here's two different mammals. So this is a mouse. Alyssa wants to get in. Can be introduced to some weird looking animals. So this is a mouse, and then that's a possum. Okay. So one of the things that Darwin was thinking about when he was thinking about development was the growth rates. So changes of structure at an early age will generally affect parts of subsequently developed, and there are very many other correlations of growth, the nature of which we are utterly unable to understand. So hopefully we're able to start to understand these things now. Okay, so two main groups of mammals that I want to look at and try to get a sense for modularity patterns in. So marsupial mammals, placental mammals. So there's another group of mammals, monotremes, but there's not many of those. So that's why I've mostly retained my sample just within here. Okay? And so I want to look at both modularity, then also something called disparity, which is morphological variation. So are those two things associated? Okay. So marsupial mammals, they have this much, much different reproductive strategy compared to placental mammals, right? They're born really, really early, they have to climb to the pouch, and then from there they suckle. So I want to get a sense, I want to poll the room here. So this is a dunnet. So I want you to tell me at which point do you think along here the baby dunnet is born? So do you want to? So let's go. So 0 to 4. Who thinks it's from that? So 5 to 9. Half-ish. 10 to 12. And 13 to 15. Okay. So 0. So a dunnet is born when it looks like this, like a little jelly bean. And so when a dunnet, so this is to scale. So this is the size of a dunnet marsupial baby. This is the size of a mouse baby when it is born. So obviously massive, massive differences in size. And it's just a consequence of these massive differences in reproductive strategy. So marsupials are doing something super different. And so when we were just thinking about how Darwin was talking about these differences in growth rates, the growth rates in marsupials are super, super different to the growth rates in placental mammals. So what do they need to do? Well, they need to be able to climb. So they've got to be able to develop these massive forelimbs, grasping forelimbs, full sets of digits to be able to hold on to the fur and climb up to the pouch. They've also got to be able to have these massive heads because they've got to be able to suckle. They've got to be able to bite down and suckle. And so from then, they would stay within that pouch for a long period of time and develop within the pouch. So they are essentially specialized within those features that allow them to do that very early transition into the pouch. So they have huge forelimbs and huge heads. And so that's super different to placental mammals, right? And it sets up this really interesting constraint idea. So you don't see any aquatic marsupials. There's no whales. You don't see any marsupial mammals that are capable of true powered flight. There's no marsupial bats. Okay? And it's because of this constraint, this reproductive constraint that's put on them at the very start of their lives. Here's these bat wings starting to form. That isn't going to be very good at grasping and being able to climb up to a pouch. That head isn't going to be very good at being able to suckle. That certainly isn't going to be very good at being able to grasp onto fur. Okay? So you don't get any kind of prosodactyle or artidactyle style things. So there's huge amounts of modifications to placental mammal limbs, huge amounts of modification to placental mammal skulls that just aren't possible in many marsupial mammals. Okay. And so I tried to ask the question essentially, is that because of modularity? So integration is just the correlation among traits, and modularity is the pattern. And so here we have our mammal jaw, and you can basically think of it as broken up into two different regions. You have corpus, so where all the teeth are, and then the major muscle-bearing region, which is called the ramus. So you have a big muscle attaching it to here, and you have some other smaller fibers attaching onto these different processes here, and then that's the jaw joint just there. So we can use the same mathematical approach, statistical approach that Sarah was using, so you can use morphometrics, putting a load of landmarks on there to start characterizing the shape. And what you can start to get a sense about is how correlated are some of these pairs of landmarks. So 10 to 12 and 8 to 11 are somewhat correlated. 6 to 12 and 11 to 12 are really correlated. There's not really any correlation between 11 and 4 and 4 and 2. And there's some pretty strong correlation between 4 and 2 and 5 and 3. And so you can start to get a sense about using these correlations, there appears to be differential growth in those two different regions, in the muscle-bearing region and in the tooth-bearing region. Okay. So I want to get a sense for those patterns. Right? Do they vary between marsupial mammals and placental mammals? Do they have different patterns of these suites of traits that are associated to each other? And so the expectation is that because these marsupial mammals have to suckle at such an early age, their heads, their limbs, they all have to be able to be operational at such an early age. Those patterns are going to be really based on where muscles are attaching and where the teeth are. So these kind of more functional biomechanical style modules. Whereas placental mammals, they don't have that same constraint. So maybe they're going to be more modular. There's still muscles that are attaching and still muscles that are developing, but they don't have to be fully functional at a really, really early embryological stage. And so their patterns are probably going to be more reflective of both biomechanical and other cellular processes that are occurring. So you can really get a sense for it. This is only a few weeks old, so those mechanical environments that they're developing within are super different. And so you're probably going to see these kind of strong uniform forces that are applied across the whole jaw, whereas at the same stage, with these pigs, for example, it's going to be much weaker. They're not going to have that same kind of biomechanical pressure. So I used the same kind of analysis. I wanted to run a principal component analysis. And so this allowed me to get a sense for where my marsupial mammals and my placental mammals are all sitting. And each of those, I guess, ends of the axis are represented by these 4 different groups. So here's the Tasmanian devil. This is called a falnok. This is a Madagascan carnivore. So those carnivores are really, really strange. So that's like the fossa, those kind of groups. There's only like a handful of them that reside on that island. This is a kinkajou. So this has a really, really short face, really strong biforce. It's biting into like big, hard fruits. And then this is an otter. And so you can kind of get a sense for the morphological variation within these two groups, right? Huge amounts of morphological variation in placental mammals, a little bit less in marsupial mammals. And so when I start to get a sense for the correlation patterns across these different groups, marsupials exhibit this kind of model. And so they have often the processes that are bearing muscles are often associated with each other. There's the jaw joint process also represents its own module, and then the whole of the tooth-bearing region represents its own module. So we've got fairly low disparity, so fairly low morphological variation, and then fairly low modularity. And this is in contrast to placental mammals. So they have really high levels of modularity. So you've basically been able to split it up into two separate modules for where the two different sets of muscles attach to, the jaw joint, and then it's often splitting its jaw into two sets of different modules as well. And it represents these different sites of cellular condensation. Okay, so in the last 5 minutes or so, blast into some cichlids. Okay. So one of the questions that I'm super interested in is looking at differences in jaws. And so luckily for me, fish have two sets of jaws. So there's the oral jaw here, and then the pharyngeal jaws. So those are modified gill arches that sit in the throat. And they have been able to separate the processes of prey capture from prey processing. So theoretically, the oral jaws at the very front of the mouth should be able to do whatever they want. The jaws at the very back should also be able to do what they want. Okay. There's this functional modularity between them. One's going to be processing and one's going to be capturing. And so what I wanted to do was essentially characterize the shapes in these two different jaws and see if that was the case. Right? So we can get a sense about how similar are these two different traits to each other. And then one extra thing I can do is start to get at the genetic basis of these different traits. Okay? So which genes might be involved in coordinating the association between these two different jaws. So I measured a whole bunch of cichlids to try to get a sense for how associated each of those different jaws are. And so what basically happens is that all the jaws, all the individuals that fall out down here have these really, really thin pharyngeal jaws and these really, really thin and long oral jaws. The ones up here, this one here, these have these really, really wide and robust oral jaws and really wide and robust pharyngeal jaws. And so this is what it looks like when you're like sort of sitting right on that line. So this is Trophiops. We've seen this guy a few times. So this has fairly wide-ish jaw with a fairly wide-ish pharyngeal jaw. So Labio is doing something a little bit different. This has an insanely wide jaw, but it has a really, really thin and slender pharyngeal jaw. And it's sitting right here, kind of off the major part of that axis. So it's been able to start to dissociate those two different morphologies. Right? And indeed, when we kind of measure them together, Trophiops has this pretty tight association. Labiotrophius is a little bit all over the place. So there seems to be that the association is much weaker between those two groups. Right? And so if we cross these two, just like with the pigeons, you can start to get a sense about perhaps what the genetic basis of these two different traits are. And so when we cross them, we do see the associations pop in again. And so if there's a phenotypic association, does that mean that there's also a genetic association? Is there some gene that's controlling the morphology of both the oral jaws and the pharyngeal jaws, or variation in those two? And so we can turn to this method called quantitative trait loci mapping. So what that means is that you take a huge population of hybrids that differ in some kind of trait. So the association between your oral jaw and your pharyngeal jaw is our trait. And we want to see, are there any regions of the genome that are associated with our trait? And so each of these little red dots represents a marker, so that's where there's a nucleotide change along along some kind of chromosome, we'll just say chromosome 7 or something like that. And that if you see a peak, that means that there's some kind of difference going on. Okay? So in your hybrids, there's some kind of trait that you've been measuring in which if your hybrid has one genotype from one set of parents, it has a really large trait, for example, and if it's in the other one, if it has the genotype from the other parent, it has a much smaller trait. And so we can then say, oh, that region's probably associated with our trait. Okay. So then what we can start to do is say, oh, well, our gene of interest must be somewhere within this region then. Okay. And so we'll do this across the entirety of every single chromosome, and maybe there's going to be no genetic overlap between our oral jaw and our pharyngeal jaw. But if we see some kind of genetic overlap, that means that there might be some kind of pleiotropic effect. There's a single gene that's impacting both of those traits. And we see that. So this is when I ran it, and we get overlap between regions of the genome on chromosome 7. So that's basically saying that there's some kind of association between genotype and phenotype for both our oral jaw and our pharyngeal jaw. So you've got this evidence for pleiotropy. Single gene impacting likely multiple traits. And so when we kind of take a look at this region, there's one particular SNP that resides in this region. And it sits right between these two different genes here. So demeclin is a cartilage gene, and SMAD7 is a bone inhibitor. And this is, the SNP is totally different whether you're a labiotrophius or whether you're a trophiops. And so if we look at expression of those two genes, they're both different in the oral jaw between our two different species. So SMAD7 is an inhibitor, so that's why the things are reversed there. But then when we look at the associations in that expression, we see associations are really, really quite tight for SMAD7, whereas they're less tight for demeclin. And if we look even closer, what we see within this region is that labiotrophius has this pretty high amount of variation in the expression of SMAD7 in the pharyngeal jaw, whereas trophiops has this pretty tight level of expression. And so if we cast our mind back to that original plot where we had our trophiops and our labiotrophius, one thing that might be going on here is that SMAD7 variation in the expression of that gene might allow them to independently vary, at least in labiotrophius, independently vary oral jaw shape from pharyngeal jaw shape. Okay. So you can start to work it in the same framework. Right? So here's your different structures. Here's the association between my oral jaw and pharyngeal jaw. And if I put in SMAD7 here, it seems that SMAD7 is able to regulate associations between pharyngeal jaw and oral jaw. But labiotrophius must be doing something a bit odd, right? Because it's able to do kind of whatever it wants. It's able to have a really robust oral jaw and a fairly slender pharyngeal jaw. Whereas trophiops has that much tighter association between those two different jaws. And so we kind of want to figure out what's going on there. Okay. So to kind of finish off here, I'm sort of always kind of interested in like what would Darwin do first here? Would he want to look at evolvability in his finches, or modularity in his Galapagos turtle shells, or just start and do a load of genomics on pigeons? Right? And so I think this, in my opinion, is probably one of the most poetic pieces of writing of any kind of science writing. Here is grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed laws of gravity, from so a simple beginning, endless forms most beautiful and most wonderful have been and are being evolved. So this is his endless forms most beautiful section, and I hope that I was able to kind of show you that through domestication, through natural selection, through sexual selection, that there is a wild amount of forms. And we're starting to kind of be able to unearth how and why there's so many of these different forms. And so with that, I wanted to thank some of my students, so especially Sarah, helping me kind of get off the ground. DePaul for giving me some, well, for hiring me. And then for giving me some money to start up and get those initial CT scans, and then some of my initial advisors. So with that, and I guess in like 2 minutes, I will take any questions.
There's a question.
Yeah.
Hi, very nice talk, very interesting. Of course, this is a completely different field than mine. I have two questions actually. So first question, I think some recently there have been studies showing that, like not studies, reports that showing that humans' jaw is getting smaller, like compared to, let's say, 15,000 years ago or a long time ago. So I'm wondering if that's kind of fit your modulatory model, and then I'm just wondering for the fish, do you see this kind of, just within the same species, do you see this kind of change of the jaw size or structure over time instead of comparing across species?
Well, so I'll answer you so the first question. So, yeah, right. So I, this is the whole reason why most of us have to have our wisdom teeth taken out, right? That we're probably not eating, well, clearly not eating the same foods that we were eating, 5, 10, 15, 20,000 years ago. At the turn of the kind of the agricultural style, when we kind of left those sort of like nomadic ideas and went to sort of more subsistence farming, we were probably changing a lot of the things we were eating. And so my assumption being that whereas previously we were eating a lot more hard foods, we're now eating a lot softer foods. Right, like if you're trying to pull a load of meat off of a bone, that's probably a lot different to just like eating a piece of pizza. So this is trying to get at these ideas of like, you need to have both the genetics, but then you also need to have this mechanical feedback. And without that, you're not going to develop a kind of a normal jaw, whatever that means for natural selection. And then what was your second question? So about cichlid growth rate.
This is actually all part of the first question. My second question I haven't asked yet. So the question, well, maybe the real question is that since we observed that in human, I'm just wondering if there's any studies showing that in fish within the same species, same kind of fish, do people see this kind of change in jaw size or jaw structure over a long span of time? I know you compare
Like through development or just among species?
Not among species, just the same. We think just any species of the fish, let's say, if there are plenty study, if you compare now to, let's say, like thousands, tens of thousands of years ago, I don't know if there's such a data or information out there. I mean, for human, we can see, we know, our diet change. Fish, I don't know if they're in the water environment, if things change much for them, maybe there's not change.
No, so the couple of examples that I point to there is like dam building. So I had some friends that there's a really good collection at Harvard in the Agassiz where they were able to get cichlids from a region on the Amazon that had not been dammed, so from like 70 years ago, and then they went down there like a few years ago and resampled all the populations of the same species, and you see these massive changes, right? So like you can imagine the difference between like fish that lived in a river versus now fish that essentially live in a lake, and so there's these massive differences in 70 years, right? Where you've just got these, often what they've done is they've, because they're living in these much more sedimentous environments, their visibility is way lower, so they've like increased like their eye sizes. So their eyes are much, much bigger, their heads are much, much deeper to accommodate that. They eat slightly different things, they eat way more like invertebrates than they did like 70 years ago. So we can, even within, I don't know how many, like 100 to 200 generations or so within these fish, we can see differences, and because of like human impact.
I guess so. Darwin, he used a lot of species and studied the ecosystem in the Galapagos, and I was wondering if you could drop him anywhere in the world today, what ecosystem would you want him to study? Like North America, in the ocean, where would it be?
So I mean, obviously, I'd want to drop him in Malawi to let him see all the cichlids. But I think probably some kind of islands, right? Like, I think one of the reasons that he loved the Galapagos so much is because you get these really bizarre animals that evolve, organisms that evolve in these kind of more isolated areas. And so, like, one of the groups I was talking about there for mammals, right, was these Madagascan carnivores. And they look really, really different, right? Like, if you were to plot Darwin in Madagascar, you know, you have the baobab trees, you have these weird, like, Madagascan carnivores, you have all these, like, endemic species of bats. Any kind of island, I think, if you put him on one of there, I think he would really enjoy himself.
Maybe one more question?
I guess, is there fossil evidence in which what like the ancestral cichlid morphology looked like, or what like the common ancestor cichlids, what their jaws might have been shaped like?
So I've got two things about this. So we do have a pretty good sense about, like, how cichlids came about. So, like, basically when Gondwana split, there's two ideas. Like, one is you have the South American group and the African group, and they were just separated. That's one idea. And then there's also the idea where it started to drift apart, and then you had these, like, intermediate forms that could basically live in marine environments. So we're not quite sure which of those is accurate. So there are kind of, like, transitional forms, but at the level that I'm looking at, no. So basically these lakes are really, really recent. That's one of the reasons why they're so exciting, because all of these speciation has occurred within the space of, like, 100,000 years to, like, a few million years, especially in, like, Lake Victoria. That's potentially like 16,000 years. And so we just, the fossil record for them just isn't really there for these more recent divergences.
I got you, thank you.
Okay, thank you, everyone. Let's not go yet. A couple of tokens of our appreciation. One is we give our Darwin Day speakers an official certificate, and it includes one of the figures that you used in your talk.
There it is.
Yeah, we planned that perfectly. So I will
Actually, hold on.
hand it, yeah, so we get done.
Yay!
And then the other thing we have is we have an anonymous donor that's giving you a $100 gift certificate from Amazon, so you can go to visit Owen.
Thank you, everybody.
Thank you. So we have some posters set up outside, and then there hopefully is still some food left outside and some drinks. So, you know, feel free to stay after the talk to each other, ask Dr. Conith more questions, and thank you for coming. And we also have a snake in the front.
Oh, yeah. That's what he's been glad. You have like an observance. Did you say so?
To be honest, I've had drunk arm.